I'm here today to present on some work that we've done looking at the metagenomic study of species diversity in UK food industry biofilms. So we carried out a project. I'm having issues with my slides here. Whoever. So we carried out a member-funded project which looked at the inactivation and removal of bacterial biofilms in the UK food industry. And as part of that project, what we wanted to do was to find out for ourselves, for our own purposes, for the experiments, what sort of species were abundant in UK food industry biofilms. So we set out to take some samples from different UK manufacturing facilities and to analyze them to see what type of species were present so that we could then use that to inform some later experiments that we did looking at inactivation and removal. So what we did was we set up factory visits over a 1-year period. We visited a total of 7 factories. And we sampled various areas for biofilms. And we used a combination of traditional methods, so plating, MALDI identification, etc., and metagenomic analysis to try and determine the species composition of these biofilms. Excuse me. So the sample was carried out at 7 processing facilities. They covered a wide range of different facilities. Mainly, they were chilled facilities. So we looked at 2 meat facilities. One was a cooked meat facility, a deli, and the other one was a raw meat processing facility. 2 ready meals facilities, 1 smoked ready-to-eat fish facility, a dairy desserts facility, and finally an ambient pH stable retorted product manufacturer. So one of the first challenges that we found was actually finding biofilms in the first place, because it became obvious from our members that a lot of people were interested in finding out what microflora were in their factory, but they're at a loss as to how to actually find biofilms in the first place. So what we used was a couple of commercially available products, one of which is called BioFinder, and is produced by iTram Hygiene, a Spanish company, and distributed by Freedom Hygiene in the UK. And this is basically works on the principle of catalase. So a lot of catalase positive organisms in biofilms. And this is a stabilized orange gel, which you can spray onto surfaces. And in the presence of catalase positive organisms, you'll get a very fine white bubble reaction, which very clearly demonstrates the presence of catalase positive organisms. We also used a product called TBF 300, which is made by Christine's Food Hygiene. And in this instance, is more like a disclosing tablet. It actually reveals the presence of bacterial extrapolymeric substances, and it stains them a bright pink color. And you then obviously can wash them away, and that will remove the bright pink color. So we use these 2 products in combinations which have slightly different mechanisms and allowed us to target areas which were likely to contain biofilms. So each facility we visited on 1 occasion during a routine hygiene shutdown, and we sampled post-hygiene, so after cleaning and disinfection. Sample sites we identified using these 2 products, and we selected food contact where possible and non-food contact areas as well. So we had a good appreciation of what organisms were present in both of those scenarios. To try and improve our recovery of cells, because obviously biofilm is very well stuck to surfaces and sampling can be quite difficult. Instead of just using routine cotton swabs, we used a combination of cell scrapers to get a bit more abrasion to remove the biofilm from surfaces, and flux swabs to allow us to get a little bit better recovery of biofilm from the swab itself so it doesn't become stuck in the cotton matrix. But still, the recovery is probably not 100% perfect, because it's very difficult to 100% remove a biofilm from a surface just by swabbing. So each of these samples we returned to the lab and we recovered any biofilm from the surface by a combination of vortex mixing and then sonication to try and break down any clumps of biofilm that were still remaining in suspension. And we tested the swabs for various different things. So the first thing we did was we tested total viable count, which is a plate count at 30 degrees C. And obviously we'll give you every organism that's capable of growth at 30 degrees C under aerobic conditions. And we then isolated the main 3 colony morphologies that we found from each sample, and we identified them using MALDI biotyping to give us an idea of the sort of organisms that we could culture from biofilms, and also to give us a wide range of biofilm isolates that we could use in further experiments. We also took each of these isolates and we carried out biofilm forming potential experiments using crystal violet microplate assays. So each of the isolates was grown, it was applied to a microplate well, and it was incubated, and then it was tested with crystal violet to stain to see whether or not it was a biofilm forming organism. And we ranked those from weak, medium, and strong, depending on the absorbance obtained on a microplate reader. Additionally, we tested the swabs to look for levels of carbohydrates, proteins, and uronic acids as potential components of extracellular polymeric matrix. And we used the methodology that's reported in Mays et al. In their 2017 paper, looking at a similar subject. And finally, we took the swab suspensions and we underwent metagenomic analysis to generate microbial profile, bacterial profiles for each sample. So looking at metagenomic analysis, natural microflora has traditionally been a difficult area to assess because, of course, the techniques that we use actually select for specific groups of organisms. So to get an appreciation of the overall microflora of any situation, of food or an environmental surface, is very difficult because every cultural step you add is going to add a little bit of bias towards certain organisms that are better at growing at under those conditions. So for example, if you look at a traditional total viable count carried out on plate count agar, you're going to select slightly obviously for the organisms that are capable of aerobic growth, firstly. Organisms that are better at growth at 30 degrees C over 2 or 3 days will predominate over organisms which may be less good at that. And also the nutrients available in any media are going to select certain sets of organisms. But it will provide you obviously an estimated count of microorganisms on any given sample. Metagenomic analysis, or as we're calling it, advanced microbial profiling at Campden, looks for the bacterial DNA in a sample and will provide analysis of the types of microorganisms present. So the type of metagenomic analysis that we used looked at the 16S ribosomal RNA subunit, and that gives that's highly conserved within bacterial species, and so it can be used to identify bacterial species present. So this technique gives not only the organisms present, but also their relative abundances. So it allows you to build a microbial profile for each sample, which you then can compare between different samples to see what the comparison is. So a typical example of the sort of thing that we've been doing in the past with this type of technology at Campden is things like spoilage investigations. So for example, in modified atmosphere packed cooked chicken that had spoiling packs with a blue discoloration, we carried out some metagenomic analysis to compare the bacterial species present in each, or genera, sorry, present in each pack between a normal chicken and a spoiled blue chicken. So on the image here, you can see on the left-hand side, you've got a stacked bar chart, which shows the genera present in normal chicken. On the right-hand side, you have the genera present in spoiled blue chicken. And the size of these bars represents the relative abundance. So as you can see in normal chicken, it has a large abundance of Faunobacterium and of Psychrobacter, which are typical organisms you might expect to find on chilled foods, particularly chicken and meats. But in the spoiled sample, we were seeing a much larger level of Pseudomonas, obviously a slime-forming organism, and a pigmenting organism as well, and of Arthrobacter, which is just a hygiene indicator organism. So it's a very useful tool to determine the changes in a product microbially, and to associate those with perhaps a spoilage symptom, or to try and predict shelf life. So the metagenomic analysis that we carried out on our swabs was done externally. The DNA was extracted, and the 16SR DNA was sequenced using an Illumina MySeq system. bacterial species present in each sample were identified by comparing against the public database of bacterial 16S sequences. And then finally, we analyzed the data using R software and the phyloseq package to compare the bacterial species composition in each sample. And that gives us a profile for each swab sample. So as an example of the type of output from that type of analysis, here's an example of one of the swabs. So this particular swab was from a biofilm sample that we found behind a seal in a forming tool within a ready-to-eat fish factory. And as you can see, the genera found in that sample are listed down the side, and each color represents a particular genera. So for example, here, Clostridium is represented by a green color, Luteococcus by a light blue color, and Gluconocetabacter by a sort of greeny bluey color. And so you can see the sort of relative levels of different organisms there. So for example, here, Clostridium species, or Clostridium genera, formed about 50% of that particular sample. So it gives a lot of information about the bacterial content of a particular sample. So you can generate a lot of different data with this, because obviously it's a very powerful tool. It will give you the not only the species present in any given sample, but also their relative abundances, and you can use that for all sorts of different things. So in this example, we have ordinate analysis, which essentially allows you to see how similar groups of samples are to each other. So for example, you might want to know whether biofilm samples from a fish factory are similar to that found in a meat factory. And this will show you roughly how close they are in terms of data sets. So as an example of that, and some stuff that we've done in the past, this is really demonstrating the selectivity of different growth media. So we have here on the right-hand side in this ordinate analysis graph, we have samples that have been extracted directly from a beef burger. We have samples that have been extracted from a PCA plate, which has been cultured from a beef burger, which is a non-selective media used for total viable count. And we have samples that have been extracted directly from VG agar, which is selected specifically for Enterobacteriaceae. And as you can see, they don't cluster together at all, which just demonstrates the selective nature of the media culture methods used. So coming back to our factory swabs, the TVCs we're finding, and bearing in mind this was post-clean samples, albeit samples that have been targeted specifically to areas that contain biofilms, so might be expected to be a bit higher, varied quite a lot between different factories that we visited. So for example, at the meat factory, we found very high levels post-sanitization in both food contact and non-food contact contexts in biofilm samples. In the ready meal factories, slightly less, but in some of the food contact scenarios, we found quite high levels, up to 7 logs per swab. And in the fish, desserts, and delis factories, we had progressively less. And certainly for the deli factory, we found several below the limit of detection swab samples, but we still got bacterial profiles for. So looking at the data that we generated, we can start to see how many different individual organisms have been identified within each sample. So this is a representation of that. So the scale up the left-hand side there, up for diversity, is a measure of how many different organisms have been identified within each sample. And then, of course, the colors represent the different factories here, and each of the points is a specific sample. So as you can see, we did find a lot of species in every biofilm sample. It's in the order of hundreds. In the ready meal factories, we found probably the most, and in certain cases, we're finding thousands of OTUs in each swab sample. So it just shows that it's very diverse populations in food factory biofilms. So if we look at the sort of order level at the different profiles that we've generated, we found that we get a lot of Actinomyces talis. These are soil bacteria, particularly in the ready meal factories, but actually throughout all of the factories we looked at had very high levels of Actinomyces talis, particularly Arthropacter. And besides that, The profiles varied quite a lot depending on which scenario we were testing. So, for example, the fish factory had a lot more Clostridium, Clostridiales, and some of the other factories, for example, dessert, had a lot more diverse range of different bacterial orders. So if we take that down to genus level, we start to see a bit of a difference between factories. So we had a lot of Brocathrix species in the meat and the desserts factories. And a lot of these factories that we're looking at also are chilled factories. So we had a lot of psychotrophic organisms, so things like Microbacterium, Cocuria, and Arthrobacter we found a lot of. But also Acinetobacter, not represented here particularly is Pseudomonas, which is a bit lower in the mix, but we did find them in pretty much all of the samples that we looked at. So if we break down to the sort of individual species, we found that for Arthrobacter species, we found a lot of this genus across the different sites. And interestingly, it seems to be represented mainly by a single species, which was Psychrochitinophilus, which we're finding in lots of different food factory scenarios. So particularly in the ready meals factories, but also in the meat, fish, and the deli factories as well. This is a psychotrophic bacterium, which is in the order Actinomycetales, which is very common in soil and water, but readily will contaminate foods as well, and is considered to be a hygiene indicator in terms of foods, its presence in foods. So looking at sort of probably your typical biofilm forming organism, certainly a slime forming organism, Pseudomonas species was found in pretty much all factories, less so in the ready meal factory, and less so probably in the production areas of the source factory. The representation here is mainly from samples taken from a water tower in the source factory. You can see that the main species present across the piece are Fragi and Lundensis, and together with fluorescens, these are the sort of main slime-forming organisms that you'll find as spoilage organisms of meat, of milk, of vegetables, etc. So it's not surprising to find them in food industry biofilms. And finally, looking at Acinetobacter species, again, very common biofilm forming species. These pink bars that you're seeing on each one of these are the species that aren't actually able to be identified to species level. So this is the kind of remainder, which it can identify as an Acinetobacter species, but it can't actually identify which species it is due to the length of reads used for 16S sequencing. So again, we found a certain species predominated more than others. So for example, Calcoaceticus we found a few times, and Junii we found particularly. And both of these organisms we were isolating from plates as well. And they tended to be probably the strongest biofilm formers that we were finding out of all the isolates that we obtained. So yeah, just a sort of deep dive into one particular factory. We found particularly potent biofilms in the meat factory that we visited. So just to show you some of the results, so that some of the sort of sample locations that we used included, as I mentioned, food contact and non-food contact surfaces. So we have, for example, cutting blocks, hand wash basins. We had various pieces of equipment and utensils that we used in the preparation of raw meat. And these were sampled post cleaning and disinfection. And as you can see, we got some very, very high levels of TVC post disinfection. So it ranged from anywhere between 5 logs for the plastic curtain all the way up to 9 logs that we're pulling out of some of the drain biofilms that we found. The predominant isolate is shown there as well. Interestingly, the Maldi biotyper that we used really struggled with some environmental biofilm isolates. So in this one, it's probably the best set of data we got from the Maldi. But in a lot of biofilm isolates we tried to run through it, we got no ID. Whether that's because the wild strains differ slightly from the strains that the database has been set up against, maybe the case. And within those, we found a range of different biofilm forming capabilities. A lot of them were extremely strong biofilm formers, unsurprisingly, given their isolation from biofilms. Okay, so looking at the levels of carbohydrate, protein, and uronic acids present in each sample, these vary quite widely. And looking at the data we have on both levels and also on the actual bacterial species present, there doesn't seem to be very much correlation between the levels of different entities and that data. But we did get quite a variability of different levels of uronic acids, different levels of proteins and carbohydrates across the different areas. So I think more knowledge is required there to be able to correlate that to anything else, but it's interesting information. And finally, looking at the metagenomic analysis from the raw meat facility, you can see that there's quite a variety of different species profiles there, but there's quite a lot of pseudomonads, as you might expect, but Brocathrix also predominated in quite a few of the samples. And then for the drain samples, there's quite a different profile there. So you're looking at sort of Micrococcus, Cocuria, and organisms of that nature. So there's quite a variability there in terms of microbial profile seen across the samples. Okay, so what we did next was we looked at the changes in species composition over time. So we revisited the same meat factory on a weekly basis over a 9-week period. And each of those weeks we took 10 samples. And again, we metagenomically analyzed them. By this time we had access to our own next generation sequencer. So these were actually analyzed on a Thermo S5 platform. And the sequences were compared against the Thermo Scientific curated sequence database. So this is the platform, the Thermo Scientific Ion Gene Studio S5 food protection system. And we used their database as well to analyze the data. So as you can see, the relative abundances were actually pretty stable over the 9-week period. So Pseudomonas and Acinetobacter were present in pretty much all of them, as were Serratia and Shewanella. And that pretty much stayed in the same abundances over the entire period, that with some fluctuation. So this may be quite a useful tool in determining any sort of environmental changes in biofilms over time, and potentially tying in the bacterial diversity within biofilms with other environmental surfaces to see whether any sort of step change could be a predictor of maybe food spoilage or raised TVC environmentally. So these could be monitored over time to allow prediction of potential issues. Interestingly, comparison of the data that we obtained from the initial visit to the meat factory with the subsequent 9 visits, which were done on a different platform, gave some pretty different results. So in terms of the profiles, they were very different. The actual diversity seen in the Illumina platform was much higher. So something like 1,500 species as an average for the meat factory, compared with 200 to 300 species for the Thermo S5 platform. Now, that might be accounted for by differences in the databases, where the Thermo database is a curated database, whereas the one used for the Illumina samples was a non-curated public database. So it's obvious that very careful interpretation is required, because obviously the platform used and various other things can really affect the results that you're seeing in terms of profile. On top of that, you can see a representation here of the diversity. So the Thermo S5 is in blue on that top graph there, and you can see that the diversity is between 200 to 300 OTUs per sample, whereas there's a much larger spread with the Illumina, so between 500 and 1,500. And if you do an ordination plot, you can see that the 2 sets of data don't match up at all. So you've got 2 separate, very different microbial profiles from the same factory. So just to summarize, the scraper swab samples that we took from biofilms showed very high counts post-cleaning in some areas. So this is after the standard cleaning and disinfection procedures have been applied. That shows that obviously biofilms can withstand cleaning either through being in awkward, hard to reach places or indeed through being resistant to disinfection. All the biofilm samples we assessed, we found to be highly diverse. So there are at least hundreds of species, depending on which platform you look at, to many hundreds to thousands of species. Many species couldn't be identified. This is probably due to the fact we use 16S metagenomics as opposed to whole genome metagenomics, which may have got a much better return on identification, but obviously is much more expensive to run. So it's clear that metagenomic analysis is a powerful tool to help us understand the microbiome within food factories and how this affects potentially products going forward. And I expect to see a lot more use of this tool in the food industry going on. So in future, AMP is getting used more and more widely within the food industry. So for example, hygiene, including biofilms, looking at the changes in microflora over shelf life. Its use in spoilage investigations, we've already used it for that, but I'm sure there's much more application for that in the food industry. The diversity within maturation and fermentation of products like cheeses and perhaps salamis, where a particular profile might help the flavor development. And looking at factory air hygiene by analysis of air filters and the bacterial species combinations that are found on those. So a bright future, I'm sure. Thanks very much for your attention. I'll take any questions that anyone has.
Thank you very much, Rob, for a very fascinating and interesting and thought-provoking presentation. Appreciate you very much sharing that data with us today. So, Will, I just look to you to address if there are any questions coming from the audience at this time. We probably have time for a few questions at the moment.
Sure. So the first question comes from Jeff Banks, who asked, did you compare the productivity of flock swabs versus conventional cotton swabs before starting this work? If so, how did they compare?
We did. It wasn't a giant comparison. We did some comparison at the very beginning of this, and we did find that flock swabs had a better recovery. I can't remember offhand exactly what the numbers were on that, but we were getting better recovery from flock swabs. Simply because, you know, we're recovering more organisms out of the swab itself into solution.
Thanks very much. Next question is, can metagenomic studies, it's just moved, or analysis be done on biofilm formation on microtiter plates or stainless steel coupons in the lab?
Yeah, I'm sure you can analyze anything you like. So, I mean, yeah, certainly you could do it on the coupons in the lab. We did it in factories because it's very hard to mirror exactly what's happening in a factory in the lab. We obviously just don't have that understanding yet to be able to generate sufficiently realistic biofilms in a laboratory. But yeah, you absolutely could apply it to laboratory samples.
Thank you. I think we've got time for one more. Could you please share more details of the Biofinder and TBF 300 for biofilm sampling? I would say that if you have more information about that, we can send that round after the event, because it's probably going to be quite detailed, I imagine.
Yeah. No problem. Yeah, I mean, without becoming an advert for the companies involved, they were very useful in targeting biofilms in factories, because obviously before that, you're reliant on your own eye and just looking for areas that are, you know, typically hard to clean. So they are very helpful in sort of targeting these areas and finding areas that are specifically contaminated, you know. And we did find that when we were looking at areas that maybe typically had poor hygienic design or would be hard to clean, certain parts of those areas were contaminated and others weren't. So you could swab and get nothing, but using these enabled us to target the specific small areas which were actually contaminated.